And now let me introduce the speaker. Robert Deans is responsible for regenerative medicine technology at Athersys Incorporated and its European subsidiary Regenerative, which I understand is outside of Brussels, Belgium. So Mr. Deans spends the world going between Europe and the US and the West Coast, East Coast, Cleveland, where Athersys is located, and Regenerative in Brussels, near Brussels. So Athersys is developing cell therapeutics based on adherent stem cells, MultiStems, isolated from adult bone marrow. Athersys has active phase 1 and phase 2 clinical development activity in acute myocardial infarction, stroke, ulcerative colitis, and for adjunctive therapy of allogeneic bone marrow transplant. Robert is also chairman of the ISCT Commercialization Committee and serves on a number of regulatory advisory committees. Robert has more than 20 years of experience in stem cell therapeutics, having previously served at Osiris Therapeutics as VP of Research. He was also previously director of R&D at Baxter Healthcare, where he developed biological components of the Isolex 300i hematopoietic stem cell purification platform. In addition, Dr. Deans has served on the faculty of the USC Medical School from 1984 to 1992. He holds degrees from MIT and the University of Michigan, and he has done postdoctoral training in molecular immunology at UCLA. And with that, please join me in welcoming Dr. Deans to the podium to deliver his speech.
So I think we're all very excited about the forefront that we're on in the context of regenerative medicine and cell therapies, particularly to take advantage of some of the current science that's moving forward in stem cell biology. I am here to represent a company that's heavily involved in adult stem cell technology, and as a publicly traded company, I need to show you a forward-looking statement for discussions that we'll hold. I first wanted to introduce to you where we stand in our own clinical development path so you would understand some of the broader industry statements that I'm going to make later, and in addressing some of the regulatory and commercialization factors that are needed to bring these medicines to standard of care. So the simple illustration at the top here identifies how we isolate our stem cell product. The cell type we use is isolated from adult bone marrow. So as I was mentioning, I work with a company that is centered in Cleveland, Ohio, and the company works with an adult stem cell product. The cell type we isolate is obtained from adult bone marrow, so it's not fetal or derived from embryonal stem cells. The cell type is an adherent stem cell. It has a morphology very much like a fibroblast, for those of you that have had a background in cell culture. And under the specific conditions we employ, it can be amplified many times ex vivo. So we have a way to make a considerable number of clinical doses from a single donor. And I'll walk through some of those numbers in a bit. We've used the cell type in a number of clinical settings. And what I'm showing you here are 2 phase 1 clinical studies that we've accomplished. One, using the cells in allogeneic bone marrow transplant. We eliminate the complications of using an allogeneic or non-patient product in the transplant product. And as well, we've used the cells in a phase 1 study in acute myocardial infarct, so we can deliver the cells into the heart directly to the region of heart muscle damaged by a block in blood flow and limited oxygen, and rejuvenate that region of the heart. And I'll show you some of that clinical data as we go forward. As well, we're in phase 2 clinical testing using the cells in ischemic stroke, and I'm going to show you some data there, as well as some data in animal models we have using the cells in multiple sclerosis and spinal cord repair. And then we're also active using the cells to treat inflammatory conditions in the bowel, in ulcerative colitis. So we're considered to be at the mid-phase clinical development. So it was a very good introduction to this space with the reference to the Nobel Prize coming forward for generating pluripotent stem cells. And one of the reasons we're excited about that is beyond the science. It's the potential to deliver some curative therapies, which until now, at best, are only open to treating symptoms. But one of the problems we have bringing these to standard of care is understanding that they can be practiced they can be practiced economically and can fit into our payment regime and reimbursement strategy in our healthcare system. And our healthcare system is very strained with the baby boomers getting older. You know, witness one at the podium telling you the obvious. And so, yeah, it's very clear that we're going to put a very significant strain on our health care expenditure programs as time goes forward, and due to complications from disease and an aging population. And this is just showing you the cost per capita that's projected per age group, and you can see that as you age and hit 75 and older, the cost per person triples and gets very, very extensive. And this is all data that's been taken from a foundation called the Alliance for Regenerative Medicine, which has a very good website describing both stem cells and regenerative medicine therapies. So where are all those healthcare dollars going with those rising costs? And you can see that the majority of those are going to Medicare-driven hospital healthcare expenditures, and that it's expected to rise to 16% of our GDP. You know, the take-home message here that I'll reiterate during the discussion is that we can't afford to bring these new therapies out to the people that need them, and we have to be very creative in understanding how to manage the economics of this emerging scientific area. So this is a slide that's generally used at meetings I go to talk about investment capital coming into developing therapies. So my background is in molecular biology and immunology, and probably 20 years ago, when I left academics, I left basic science, and my interest and my rewards became very applied. And it's something that I've enjoyed, and one of the reasons is, and I'll tell you, there is no reward greater than having worked on a product that enters the clinic and is used in man. I think the fact that you can make something that's not harmful and you de-risk and have a safe therapeutic product come forward is one thing. But applying some of the creative scientific technologies that I'm going to talk to and seeing those move into man is incredibly rewarding. And so for many years, my values were really driven around this movement from preclinical development into phase 1 clinical testing, first entry into man. So that's all well and good, but somewhere maybe 5 or 6 years ago, I realized it's not just about making these technologies work, it's about my own need to have these available when I retire and need treatment. So, you know, this is serious, and being in a position to influence, it makes you very motivated. So my motivations really moved from entering the clinic to understanding how these could be generally available to the public, and what's required to progress through clinical testing, and what you have to know in addition to science to make this a reality. And so I think most of you are aware that clinical testing and product approval, bringing something out so a physician can use it without, under prescription, can use it openly, goes through 3 phases. A phase 1 clinical study where you demonstrate safety, so the FDA regulates the product in a small clinical study, so you demonstrate safety. You then move into a phase 2 clinical study, which involves setting the dose parameters and understanding what the clinical positive readouts will be in order to take the study forward further. And so when you have that hint of efficacy, you know the dose regimen and the dose that is responsive, you move into a phase 3 clinical study, which is essentially setting a statistical response. It's a gamble. You say, if I give a patient this product, I'll see this response in a disease setting, and you tie it to a statistical basis. If you hit that, the FDA approves the product. If you don't hit it, it fails. And the difficulty is it takes an incredible amount of capital to move through these, all the way through this clinical development cycle. And so one of the things that you also realize is that to get a product approved and out to standard of care, it's not just about science and it's not just about the FDA. It takes a village. There are a whole number of people that are involved here. A company like mine would be considered the therapeutic sponsor, but a lot of the decisions we make about developing a therapy are driven by patient advocacy groups. We know what new therapies people are interested in. We learn where current standard of care is non-existent. There's significant unmet need. So we may make a decision to pursue a specific disease indication. We interface with the FDA or the European regulatory agencies that tell us how to do that in a guided way for patient safety. But we do that, and we can only do it with quality if we can engage the translational physicians, the medical community, to undertake the clinical testing. And when the product's done, they're the people that are going to decide whether it has significant benefit, and they'll prescribe it to the patient. So it's very important to work in a very engaged way with the translational physician community. So when you're done and you have FDA approval, it's another story to set a selling price for the product. And that's done by negotiating with private insurance companies or Medicare to set a fixed price for the product. So that price has to be something that insurance companies are willing to pay in order for this to be adopted. And then all along the way, you're negotiating with the capital community, venture capital, investment banks, or you're negotiating with pharmaceutical companies or healthcare companies in order to bring capital in for that expensive clinical development. So I just wanted to take a slide and explain two basic paradigms in cell therapy and show you how they reflect where we began with bone marrow transplant and where it's taking us in terms of personalized medicine. And so it's very timely to talk about this because it was really, I think, about 2 weeks ago that Dr. Donald Thomas, the pioneer of bone marrow transplant, passed away. And so this whole area of regenerative medicine began through the practice of medicine by a physician that decided that he could manage complications of bone marrow transplant, that in cancer patients who were treated with myeloablative drugs, that the immune system could be restored by either an autologous or an allogeneic hematopoietic stem cell product. And so we're all familiar with solid organ transplant, lungs or livers, etc. And bone marrow is basically a tissue transplant. And so the paradigm there that we're all familiar with is that transplants are patient-specific. You get one organ or one tissue, it goes to a patient, it has to be immunologically matched to the patient to avoid immune complications. But it's really that concept, that paradigm of one product for one patient that drove out this industry. And it was followed by other immune cell types, therapies with other immune cell types like T cells, engineered T cells, or people have used bone marrow to treat other conditions than immune complications. People have used bone marrow cells to treat peripheral vascular disease or stroke or other cell types. And that's what I refer to as heterologous hematopoietic stem cell use. So the cell type that we work with is in a class of cells called the mesenchymal stem cell. And there are properties that this cell has that shift this paradigm for treatment. And it takes it out of a transplant product paradigm with a tissue designated for a single patient and moves it into a biologics paradigm where the cell product can be used without immune patient matching against immunological parameters, and it can be used off the shelf. So it can be used like an antibody biologic, or it could be used like a drug. So this had very significant impact on development of this space. And before I turn to the reasons why, I want to let you know that the technology that we're seeing with pluripotent stem cells and a rise in tools for analysis of epigenetic or genetic changes in cells is returning us to the transplant paradigm by restoring this ability to practice personalized medicine. That is, an IPS, a cell from your body, could be de-differentiated to a more primitive developmental state. And from that state, your own tissues can be regenerated and used in a transplant setting. So we're going from a tissue being used specific for the patient, and all of a sudden moving to another stem cell type that can be practiced as a biologic, but the field is going to move back to a very personalized medicine approach. So this is an illustration of the product that I described to you previously, that we isolate from a single healthy donor, the bone marrow isolated from the iliac crest of the donor. And I'll show you a description of the manufacturing process in a moment, but we can basically get over 200,000 clinical doses out of a single donor. So our ability to expand these cells to large number and their potency is a very powerful concept. So these cells do not trigger an immune response. They very actively downregulate and prevent an immune rejection response. So we take advantage of that by using these cells to treat inflammatory diseases or diseases where, like autoimmunity or graft versus host disease in allogeneic transplant, where you have an immune attack on patient tissue. We have 5 or 6 years shelf life on the produced product, and we've developed a very deep understanding mechanistically of how the cells work, and I'm going to show you some of that science as we go forward. So these were the 2 properties of this class of cells that allowed it to become administered in the biologics paradigm. And the first is that the cells are not immunogenic. They don't stimulate an immune response when they're put in the body, even with repeat administration. And as well, when they're in the body, they turn off an inflammatory injury response, or they turn off an allogeneic rejection response. And the second property that renders these cells as a biologic is their expansion ability. Because we can make so many of these cells ex vivo, or in the tissue culture setting, we have the ability to make a large number of products from a single donor. So for years, people have been trying to attract capital into the cell therapy market, but the pharmaceutical and healthcare companies have resisted taking that step. And it's because they're not comfortable with the transplant paradigm as a business model. There's never been an effective cell therapy transplant business model developed until very, very recently with Dendreon. I can talk about that after the discussion. But with the properties of the mesenchymal stem cell being highlighted here coming into play, it's driven a number of very important business partnerships that have enhanced and accelerated clinical development. So you're possibly not aware that Genzyme invested very heavily in Osiris Therapeutics, an MSC company. We have a development relationship with Pfizer. Cephalon has put over $1 billion in an MSC company called Mesoblast. Roche has made selected investments in the mesenchymal stem cell space. So finally, pharma and large amounts of capital are coming into the development processes for this class of cells. So as I mentioned, the cells are made from a healthy donor, and there is a mechanism in place with tools in the blood cell industry so that we can isolate these cells in what's called a closed system. The cells are never exposed to the atmosphere. So there are automated platforms for processing the bone marrow aspirate. We then take cells, a specific population of cells from the bone marrow aspirate, and we put them in a cell culture device called the cell factory. These are parallel sheets of polystyrene that the cells stick to. And then we're able to fill those parallel sheets with a specific culture media. We're able to expand those for a period of about 10 days in culture, an automated system to concentrate, and we can set down a master cell bank. So this is like manufacturing an antibody type therapeutic product. And then from that, we can do repeated clinical production run and reach this large number of donor products that I was describing to you previously. So the specific culture conditions that we use actually let us take these cells out to 60 to 65 population doublets. So our yields are 2 to the exponential 65. It's a fantastically huge theoretical number. And this is an illustration of the cells that we use. These are stuck on these polystyrene. This is a 2-dimensional picture of the cells attached to a polystyrene surface. And you can see they're small. They're about 15 microns in size, and they're trapezoid in shape. And they have the ability to turn into connective tissue types, and this is what's called the mesenchymal stem cell. And these are longer cells that are more fibroblastic-like and are generally grown and fill the culture plate. So our ability to maintain our cell product under conditions where we keep it in the stem cell state are what are responsible and reflected by these large population doublings. If we drive these cells towards a bone-forming cell type by driving it into a mesenchymal stem cell, we get a very limited replicative capacity. So we can make a master bank in many runs and characterize our donor quite well. You can only get 4 to 10 clinical products from a donor if you put it into conditions to isolate and expand a mesenchymal stem cell. So we obviously, people that are developing cells in this space are interested in cell characterization and as well distinguishing the cells from related cell types. And we do that by doing computational biology and bioinformatics on the gene expression profile of the cell population. So we can analyze and screen all of the genes that are expressed by our cell product. We can do that with a competing cell product or an alternate cell product, and then we can analyze that full data set of 20,000 genes and mathematically deduce their relatedness. And so it lets us sort these genes into categories. And in doing that in a blinded way, we can see if we analyze 10 of our cell products, they all cluster together and cluster differently than a related cell type. But that gene information gives you more than just a fingerprinting or pooling capability. It lets you know, based upon the genes that are different in the two populations, what markers might be used to discriminate the cell types. And so this is the clinical cell product that we produce, and I'm going to explain the colors in a moment. And this is a related or competing cell product. What we're doing here is staining the nuclei of all these cells blue, just as a general detection reagent. And we're also staining the actin filaments within the cell, the cytoskeletal elements of the cell, red. So in all of these images, we have blue nuclei and red cytoskeletal elements. And then what we're doing, based upon these differences in gene expression, are using an antibody with a green fluorescent tag on it to stain a specific gene protein product. So in the case of caveolin-1, a cell surface linked calcium binding protein, this cell type is positive, this cell type is negative. So it's a way we can discriminate the two or tell if one cell product is contaminated by the other. And similarly, a gene involved in prostaglandin synthesis, prostacyclin synthase is on in a mesenchymal stem cell and not on in the cell product that we have. So some of this is very relevant to biology of the cells, but you can clearly understand that it's also very relevant from an intellectual property standpoint to define very specifically what your cell product is, and the expression of marker sets like this is very key to that. So we've learned quite a bit about how these cells work, and the basic model is that they're a delivery vehicle. It's almost like the Andromeda strain, where you put these cells into circulation, they travel through the circulation, and they move out of the blood system and into the tissue that has evidence of inflammation or injury. So they're basically a biologics delivery vehicle. When they find themselves at this site of inflammation, they read the environment and they express a series of biological factors that prompt repair. So what you're basically doing is identifying a cell that's the counterpart of the inflammatory cell. It's the anti-inflammatory cell. And so when these cells are in a wound environment, induce wound repair or repair from ischemic injury, lack of oxygen, they act in a number of ways in parallel. They downregulate inflammation, they stop T cells and immune cells from cytolytic destruction, they block apoptosis or cell death in the brain, in the heart, and in other tissues. They aggressively stimulate new vessel formation, so in tissues that have been blocked, like in heart attack, where you have an ischemic event and your heart tissue has no oxygen, they drive new blood vessels very quickly in a 2 to 3 day period to grow back into that region, so you rescue the heart tissue. And they also recruit other progenitors from the host to build new tissue. And they do this fairly quickly. They do their action in a week to 2 weeks after delivery, and they don't stick around. They're not themselves forming new bone or new tissue. They're having their influence trophically. They're acting on host tissue and accelerating wound repair. So anybody that's been through the drug or biologics development cycle knows the term PK/PD. It's the pharmacokinetic pharmacodistribution profile of the cells, and it's basically addressing the question of where does your clinical product go and how long does it stay there, and how is it acting and how long does it act. So it's a very obvious, to a scientist, a very obvious block of information that you'd like to know about the cell product. So unlike drugs or biologics, where you can radio label or follow the biologic or drug quantitatively in the body, tracking the cell therapeutically is very difficult. And it's very difficult in part because labeling a cell with radioisotope is only valuable if the cell stays alive. If the cell dies and the radioisotope is released, it's background signal in your study, and you're not going to get an accurate measure of where the cells have gone. So we have a very good idea of where our cells go, and I'm going to show you a way that several experimental techniques we've used to understand that. One of the ways we do that is to genetically modify the cells with an enzyme called luciferase. And this is an enzyme that can act on a synthetic substrate called luciferin. It's an enzyme isolated from firefly. And so if you take that enzyme from firefly and genetically insert it in our cells can then be delivered to the body. An animal can be fed this substrate for the enzyme luciferin, and the cells that have metabolized luciferin can then be detected. And so what you're looking at here are a series, the other advantage here is that you can analyze this signal without sacrificing the animal. So once the cells have been delivered, you can keep feeding them luciferase, and then you can tell over time how abundant the cells are they staying on that site and what the kinetics of loss are. So in this case, we've set up a model where we've injured the gut. It's a graft versus host disease model in mouse. And so in all of these animals, there's been an immune attack in the intestine. And when our cells are infused, we're putting them in rodents. We generally put them in the tail vein, so we're just putting them into the venous system of the animal. On first pass, in the first 30 minutes, they're moving through the body and they're lodging primarily in the lung and the liver, the spleen, and the bone marrow. And these are sites in circulation that are called the reticuloendothelial system, and they're responsible for scavenging material from the bloodstream. And there's a lot of phagocytic activity in these T cells, in these organ systems that would degrade and turn over a cell product. So what you're seeing here, when we've done this in 5 animals, at 6 hours, almost all of the material we detect is in the liver and the lung. This is the thoracic cavity of these animals. And you can see material at the wound site where we injected, in the tail vein, where we injected the animals. Now you can see in animals that have had this gut injury that by 24 hours, a significant number have moved out of the reticuloendothelial system and are now lodged in the gut. So they're homing specifically to the site of tissue injury. So this is like it's a smart biologic. It goes where it's specifically needed. So this is another example of that type of biodistribution and cell tracking. In this case, we're looking at animals that received an ischemic injury in the heart. We tied off one of the arteries in the heart, so effectively creating a heart attack. You have a region of no oxygen in the heart tissue, and then we've infused the cell product. And you can see that the majority of the cells are found in the thoracic cavity. This is looking at the same animal over 7 days and then quantifying the signal that we see specifically in the thoracic cavity, and you can see we get a very nice decay curve. So it's telling us kinetically that these cells are, about 90% of those cells are lost in the first week. After 2 weeks, another 90% are gone. So we're really starting to drop in cell persistence. So, just to go back to that previous slide, this is a very non-quantitative and insensitive way to track the cell population. We can only see about you have to have about 10,000 cells present in a pixel to get an image. So when we put 1 million cells in the body, we're really not able to get a very good signal over a period of time. And so we've been working with another technology called whole body imaging to give us a mass balance accounting for the cells that we put in. What we'd like to do is to be able to account for every single cell that goes into the animal and know where it goes and how long it can be found. And so the way we approach that, and right now we're running at about 70, now we can get up to about 75 to 80% mass balance accounting for the cells. So we do that by running an experiment, and then in this case we're looking at a mouse, and the mouse has been sacrificed and then frozen in a block. And then what I'm going to show you when this movie runs is that we're going to shave off slices of this animal and then take photographs as we go. And we're then going to reconstruct the photographs computationally and rebuild a virtual animal and then analyze that animal. So this is just showing the instrumentation process. This is a 50 micron slice of that animal being shaved off. And after that shave is removed, two cameras come in. One of them takes a white or bright field image. The other camera takes a fluorescent image at very high resolution. We can go from recompiling a virtual image of the animal at a gross level, or we can go down to microscopic 15-micron resolution. So this is an example. This is a mouse that has been rebuilt computationally. So this is all virtually reconstructed from those tiled images that I just showed you. And so when I click on this, it's going to rotate, and it's going to show you that these cells are lodged in the animal as I described to you previously. You can see that they're primarily in the tail vein at the site of injection, or they're in the thoracic cavity. You can see as it moves. And I'm going to show you some more specific lodging of the cells in the liver and the spleen as well. And so that's if we now, these aren't the organs from the animal. These are images that we can selectively reconstruct. And I'm showing you an image of the liver, an image of the lung, the spleen, and this is an image of the whole mouse. But basically, we can then analyze this in even more detail. I'm going to show you that in a moment. But it lets us get a very good quantification for how many cells, where the cells are that have gone in, and how long they've persisted at that specific time point. So I'm going to show you two images. This is of the liver that's been reconstructed, and it's going to rotate when I start the movie. There's a way we can specify this image to show only the blood system, and every orange fluorescent dot that you see here is a single input cell product. So this is rotating, and so these are the two lobes of the liver, and now we're dropping back to see only the blood vessels, and now we know every one of those fluorescent dots is one of the input cells that we put in. So we can do the same thing. This is in the long bone, the femur, after delivery. You can see many fewer cells, the individual dots that are there. We can localize those anywhere within that tissue image. So it's a really fascinating and fun distribution analysis that we've been able to develop. So I'm going to show you some data from our study using these cells in acute myocardial infarct. And so this is a picture or a schematic of the human heart, and one of the major arteries running down the heart that supplies blood to the left ventricle is it's called the LAD, the left lateral descending artery. And so we deliver our cells by threading a catheter down the LAD and then allowing the cells to be injected into the heart tissue. So this type of treatment often comes after an atherosclerotic plaque has blocked the artery, so you get a vessel which has been blocked, it blocks blood flow to the heart, you have the heart attack. So the first thing that happens when people present with this is that they have a catheter put down the blocked artery, and the catheter has a balloon, and the balloon blows up and it stretches the walls of the artery apart, and then a metal stent is put in place to prevent that from collapsing down again. So it's a mechanical way to open the artery. So the catheter that we use to deliver the cell product operates the same way. So you can see the balloon here is blown up in the blocked artery. But when this balloon blows up, a small needle sticks across the wall of the vessel, and our cells are injected into the tissue that surrounds the artery. And they then migrate through the tissue to the region of damage. And so this is actually an angiogram showing blood flow in the heart from the first patient retreated. And so this is the guide wire of the catheter. The catheter has been blown up, and this is, contrast agent that's being secreted out of this needle into the surrounding tissue. We do that for a minute. We deliver either 1 or 2 mils of volume. You can see 2 mils of volume have now surrounded the vessel. And now we've dropped the balloon down, so we've deflated the balloon, and then we monitor and show that we have not dissected or mechanically disturbed the artery, and you get very good blood flow back through that region. So you can see that the occluded catheter has blocked this blood flow for this network of arteries. So this is in our preclinical model in the pig, where we determined how well the cells could migrate after delivery. So the artery that I just described runs down the outside of the pig heart. We inject cells at 2 positions, and then the region of infarct is actually down lower, and then we measure how effectively the cells move to that zone of injury. And so this is just showing, this is a cross-section of this ring. And so this is the circle of heart muscle. You have the left and right ventricle. This is scar tissue that forms in the left ventricle when blood flow is blocked. And so what we're doing in this study is, and I'm not going to go into more detail with it, is showing that our cells migrate to the region right around this and induce new vessel formation and rescue that tissue and make that heart muscle functional. And so we track that in the pig studies by measuring something called ejection fraction and something called scar size. Scar size is the size of that yellow area that I showed you previously. Ejection fraction is how much of the blood in the left ventricle is pumped out. So a healthy heart can pump out about 60% of the volume of the left ventricle. An injured heart can go anywhere down, people with congestive heart failure are down around 15%. And so, an inefficient or low ejection fraction means that you're not oxygenating blood and circulating oxygenated blood out to the rest of the body. And so what we see, we're looking at in black the before injury, injured animal, and injured animal at 1 month. And now we're looking at the same treatment, but now the cells have received the stem cell product. We can see a very significant, statistically significant rebound in ejection fraction as a consequence of doing the cell treatment. And we can see that we get a very nice reduction in the scar tissue that's formed as a consequence of cell treatment. And so that's also what we've seen clinically. These are results from the patient population that we treated in our phase 1 clinical study. The patients received one of three doses of cells. These are the control group. This is a 20 million cell dose, 50 million, or 100 million cell dose. And this is the improvement in ejection fraction that I pointed to on the previous slide. So you can see where the control group falls. We get a very nice dose trend up from 20 million to 50 million, and it looks like we're hitting maximal value somewhere around the 50 million cell dose range. But it's very encouraging for us clinically. I'm showing you that data, but in purple I'm showing you the same data over a 12-month period. So you can see that the response that we see is actually very durable. The cells persist for a week, but the influence we see is very significant in terms of durability. And so this is a repetition of the data that I just showed you using our cell type. But one of the reasons we're excited about it is that a similar study has been run in Europe. It was a very large 204 patient powered study with a placebo control group as well that only showed a 2.5% increase in ejection fraction. We're up in the 8 to 10% increase range. And so long-term follow-up of this patient population showed a statistically significant in clinically relevant cardiovascular events, including an improved overall 5-year mortality from all causes. So we're very anxious to continue to develop the cardiovascular program into the next phase based upon those promising phase 1 results. So I mentioned earlier we're in a study to treat patients that have ischemic stroke injury. So you get, again, a clot or blockage of blood flow into the brain, and that ischemic limitation induces cell death. It disrupts neural connectivity and leads to long-term motor or cognitive dysfunction. So we're currently in a study. We're about halfway through treating about 130 patients. Again, double-blinded, placebo-controlled. We're using, we initially used 2 doses, 300 million, and now we're up to 1.2 billion, which have been safely administered. And we're coming in right after stroke in a 24 to 36-hour period. And we should have those results out by the middle of next year. And so obviously I don't have the clinical data to show you here, but we're very, very optimistic and enthusiastic about the approach. What I'm going to do is show you some things we've done to better understand how the cells work in terms of their mechanism of action in central nervous system injury. And so one of the things that happens when the brain is injured is that the blood-brain barrier opens. So normally the brain is protected from cell products in the body by having a very tight cell junction that regulates and limits the ability of cells to migrate into the brain. And only in conditions of inflammation or injury do cells actively move into the brain. And that's what's really represented here, that you can either with stroke or with a traumatic brain injury, the region of brain that's affected will become leaky. If you have dye in the body, you can see that dye accumulates in the bloodstream. You can see that dye will accumulate in the brain, reflecting the ability to move through this weakened blood-brain barrier. One of the things we've learned is that when our cells are delivered, they actually limit and close the blood-brain barrier in a very aggressive way. And they do that by changing the nature of the inflammatory cells that are induced in injury. But in brain injury, immediately after brain injury, you get inflammatory cytokines typified by TNF-alpha, interferon gamma that migrate into the brain, and they activate inflammatory cells. Microglia are a brain inflammatory cell that is quiescent unless it's signaled by these inflammatory cytokines, and then it acts to clear out tissue. You'll see neuronal cell loss, and you'll see the region of injury basically hollowed out as part of the healing process. And so what we've learned that our cells are doing is changing this inflammatory cytokine cascade from a pro-inflammatory injury-mediated cascade to an anti-inflammatory wound repair cascade. They do it by blocking the blood-brain barrier, and they do it by changing the expression of the inflammatory cytokines to another population, which include IL-4 and IL-10 that are known to be very wound repair and pro-angiogenic. And so I'm already getting into too many acronyms and too much jargon about inflammation and pro-inflammation, but what I'm trying to tell you conceptually is that by delivering a large number of these cells that promote a wound repair response, we limit the damage to a tissue that would ordinarily be seen by the immune cells trying to clear up the injury, and instead we accelerate repair of the tissue, including host regeneration. And that's what's going on here. And just with a couple of statements beyond that, to those of you that may have an immunology background, we know that we're doing it by inducing a class of regulatory T cells that are known to promote this wound repair and limit the inflammatory cascade. And we're also changing phagocytic cells like macrophages and microglia from a pro-inflammatory to a pro-angiogenic and anti-inflammatory response. So it's basically resetting immune homeostasis in a positive direction. So one of the other complications in stroke is that, in fact, the leading cause of mortality in the first 2 weeks after stroke is death from infection. And the reason for that is that this induction of inflammatory cytokines wipes out many of the cells in the spleen, and the patient then doesn't have the ability to mount an effective immune response. So what we've shown is that on delivery of our cells, they accumulate in the spleen. We're using another fluorescent tool to detect where our cells have gone in the injury in terms of specific organs. And we can see that with injury alone, we have relatively little signal, and then with 2 different doses of cells, we get a large accumulation of the cell product, specifically in the spleen. And when the cells are there, by modulating the immune and inflammatory cells in the spleen, they preserve the splenic mass. So this is the mass of a normal animal or an injured animal that's been treated with the cell product. This is the size of the spleen with vehicle or control treatment, injury but no cell product being used. So you can see, and we have statistics underlying the degree of protection we have, that we're getting a significant protection of the spleen and immune cells that are resident there. And so this is just more data that points to the data that I just summarized to you. And then just conceptually, I wanted to tell you that we not only look at tissue pathology and markers of inflammation, but we also do what's called in vivo tissue microarrays. We analyze on a genetic basis how the host is responding to injury, and then because we're putting human cells into these rodent models, we can detect how our human cells are changed by being in the injury site and what they're expressing that can stimulate wound repair. So we're using multiple tools to dissect out mechanistically how the cells are working. So we've also done work in models of multiple sclerosis, and I think you're all familiar that MS is a disease state, is an autoimmune disease state where cells or antibody responses attack the coating of nerve cells, the myelin sheath that surrounds nerve cells. And as a consequence, the nerve cells become dysfunctional. And so there are rodent models of replicating that disease where you immunize an animal against the myelin basic protein, against that sheath that surrounds the nerve. And over time, you get symptoms that develop over a 2-week period that mimic multiple sclerosis. So we induce disease, we wait for symptoms, we deliver the cell type, and then we carry animals out for 7 weeks total. And so there's a series of clinical scores that are developed based upon the motor function and physiology of the animals, and they're just scored here with 0 being a healthy animal and 5 being an injured animal. And then what you're seeing here is the experiment that I just described. 2 weeks prior to cell infusion, these animals were immunized, and the animals had a disability. They were scoring at about 2.5, so they were somewhere around here on this curve. And so animals at day 14 that received vehicle showed no improvement. Animals that received cell product got markedly better. And that's what you're seeing at 3 different dose ranges that we use. So we're seeing a very significant increase in motor function that we attribute to limiting the inflammatory damage that's seen. So this is just a visual of what happens in these animals when they're impaired, when they've been immune compromised, when you have the immune attack against the myelin basic protein. And so you can see the inability to carry or control the tail. You see very poor hind limb function. You see this kind of fur sloughing. That's a function of the inability of the animal to groom itself. This is just another example that is seen. And this is, I think the upper was the best case in the vehicle treated, and this is one of the worst case animals. And so I'm going to show you a very different phenotype when you look at animals that have received cell product. You can see the increased gait, the fur has now been well groomed. You can see that the tail is at times up and alert, but not limp and dragging. And obviously in the other example that we see here, similar good behavior. So it's a visual reinforcement that reflects the scoring that I had just shown you. So we know that we're doing that, we've learned that we're doing that not only by blocking the immune attack against the nerve tissue, but we're also stimulating remyelination. We're stimulating cells to come in and rebuild the myelin sheath around the damaged nerves. And it's possible, if this is a cross-section of spinal cord where we can stain for myelin with the blue dye, and what you're seeing is that in the vehicle-treated animals that had those motor dysfunctions, very significant gaps in what should be normal blue staining material. These are lesions of demyelination and loss of motor function, and you can see how much better the cell-treated animals look. And if we look by electron microscopy at myelin sheaths covering the neurons, we see a marked improvement in remyelination. So myelin surrounding this nerve is a series of concentric rings that you can see that are very deeply stained. So all of these are very good myelin sheaths that are surrounding the nerves. You can see a number of nerves here that are very heavily demyelinated, and only a few that show reasonable myelination. So for us, it's been a very nice confirmation for how these cells are working in the MS setting. So we've done some work in spinal cord injury as well, and we began that work by doing just a very limited spinal cord injury. And so this is an exposed spinal cord in a rat, and this is the injury that we caused by taking a forceps and just pinching across the spinal cord. And so it's not an injury like a contusion injury that would render the animal paralyzed. It's a physiological injury that lets us look at pathology and what's going on to the nerves that are in this region of crush. And so it's probably best illustrated on the next slide. And so this is a representation of the spinal cord, and this is the region of crush. So what happens is the crush is made, these nerves are severed, and you have a zone of injury. The green dots here represent activated macrophages. These are phagocytic white blood cells that come in to clear up the debris post-injury. And so these macrophages set down a proteoglycan scar, so you get collagen fibers or proteoglycans that wall off the region of injury. And as well, these activated macrophages cause the neurons here to pull away from the original site. So what you'd like, if you're going to treat spinal cord injury, is the opposite of that. What you want is not to see these axons pull away, but you want to see them grow back across and then reform new connections. And so what we've been studying is how these activated macrophages influence these axon ends and then how our cells can reverse that. And so let me see what So this is how we've followed that in vivo, and that is this is the injury site in that kind of crush model. And what you're looking at in purple is a stain for activated macrophages. So you can see how abundantly they've cruised into this zone of injury. And what you're looking at with red is a stain for these severed neurons. And so the severed neurons have moved back from the site of injury to this point. So in vehicle-treated animals, this is just a progressive pulling away. That's what's represented in these black bars. So here's the site of injury. Here's where at 2 days these red bodies fall. At 4 days and 7 days, they're just progressively pulling away. So in this case, we've actually come in with a green fluorescently labeled cell product, and we've injected it on this side of the injury. These cells have migrated about 500 microns to the center of injury. The green cells are our cell types, this adherent stem cell type. And what you can see is they're actually promoting growth of these red axon ends back across the region of injury. So we're getting, this is just a distribution of where the axons are in the open boxes here. These are the vehicle-treated pulling away. In the otherwise boxes here, you're seeing that we're actually promoting these axons growing back across the proteoglycan area. So it's a very, very promising observation for us about how these cells are working in that state. So we've also set up this model so we could study it in an artificial in vitro setting, and we do it by collecting dorsal root ganglia neurons from the rat and plating them on a proteoglycan scar. So we're putting collagen and laminin down to mimic a scar, and when we place the neurons on that scar, they form what's called the dystrophic body. This is not a good end of a neuron, this axon end, and I'll show you a good one in a moment, but what we understand is that when these neurons find themselves on a region of scar, they look abnormal, and then they can be induced by these activated macrophages to retreat. So the movie is going to show you, that I'm going to show you, represents that. And so we've added in a number of activated macrophages to this culture. This is the axon end. The macrophage creeps down to the axon end, and it disengages it from the neuron and causes it to retract and move back away from this field. So we've done we track a number of these neurons by time-lapse video microscopy, and we can show that they're moving progressively back away from their original end. So I'm going to show you the opposite when we add our cell product in. So this is the adherent cell type that I'm talking about. I didn't really point out the dystrophic end, but we also stimulate very good neurite and axon outgrowth by co-culturing these cells. And you're going to see a series of activated macrophages latch onto this neuron end, and a very different event take place. So this is the axon end on this proteoglycan scar. These are a series of activated macrophages that have jumped on it. But instead of mediating an effect, they're actually, you see that they're released, and now you see that based on some growth factors we know our cells are making, we're actually stimulating growth back out. Macrophages come back, and you see that they leave again. So it's a really fascinating replication from the in vivo injury model to the in vitro state. But it's interesting for another reason, in that we've been able to dissect it down to a very specific biochemical pathway. And we know that it's regulation of methylmetalloproteinases, which reform the extracellular matrix in injury, blocking their release from the activated macrophages that's responsible. So we've also looked at these animals in a more, this isn't a model of spinal cord injury that induces paralysis, but it's a more severe model that results in motor dysfunction, in loss of gait and tail use, and it also results in impaired bladder function. And so this is an animal that's, you can see, can move, but has poor hind limb function, has relatively no control over its tail. This was the vehicle-treated animal in that setting. And now this is stem cell-treated animal. So you can see much stronger capacity in tail function. The gait is much stronger. There's really a marked difference physiologically that you can see in these animals as a consequence. So for us, it's been a very exciting block of data to generate, to get down to a molecular level, a tighter understanding of how these cells are acting. And it helps us in clinical design in terms of understanding dose and regional placement of the cells and so forth. So I'm going to shift topics a little bit and talk about problems that we face in producing these cell products and bringing them into a cost-effective treatment range. And I'm not going to walk through a lot of data points here. I'm just going to tell you that right now it costs about $15 to make 1 million cells of this type. And these cells are being used by a number of companies, and the dose that's being used in different indications varies greatly. But the higher doses that are being used at producing the cells at this cost something like $75,000 to treat a patient. So when you start talking about a 100-patient clinical trial, you're talking about a whopping amount of money to put forward in order to be able to advance the technology. And so the cost goes into, at early stages, goes into primarily to facility costs. And the second major cost we have is media cost. But as we achieve some economy of scale, almost all of our costs drives down to media. So there's a complex set of technologies involved in bioprocessing that's been developed to produce monoclonal antibodies or biologics. And one of the things that's happened in our field is that the bioprocessing industry, the people that make these bioreactors and devices to process cells, don't yet believe in the clinical opportunity, so they've not developed tools for us to use to scale the technologies up. And so I'm not going to talk in specifics about the tools that we need, but most people in the industry are currently at a scale of producing about 1 billion cells per clinical production run, and are trying to get up to a factor of 50-fold greater than that. And we're not going to be able, when you get up to that level, you move away from people being able to practice this, and you move into the requirement for robotics having to practice this. And that's what I'm saying. We don't have the tools to be able to make enough cells to go into large-scale commercial production. And so it's not only the number of the tools that you need and the number of cells you need to make in cell culture, it's how you handle all the downstream aspects of getting rid of the spent media, collecting the cells, getting the cells into a vial for freezing, and then having the ability to efficiently cryopreserve the cell product. And these are just pictures of what some of those larger scale vessels are like. In each one of these trays, there's 40 racks times 4, so you get a very big two-dimensional surface area, but this is beyond any human's ability to pick it up. We get a little forklift that slides in here and moves this thing around. So these are just pictures of some of the scale-up devices that exist. So one of the problems when you're a small company is that you don't have the capital to build a large-scale manufacturing platform. And it's really a severe problem because for us to begin to do experimentation on a large scale, it's prohibitive, it's cost prohibitive. So any run that we would try to do to make 50 billion cells is going to cost us $750,000. So you can imagine if you're at an experimental phase of scaling up production and you want to try 2 or 3 variables and you want to repeat them 3 to 4 times, it's a whopping capital requirement cost. So there isn't a solution. And I mentioned that the bioprocessing industry hasn't built the tools to let this flow economically for us. And so what's happening is, which is all good, that a series of industry consortia, the International Society for Cell Therapy, Alliance for Regenerative Medicine are going out to academic bioengineering centers and pushing and promoting development tasks to go into the public domain. So people are sharing the cost of these downstream bioprocessing requirements. And so we're always pitching to people in our space to get involved and help solve some of the common production costs. And then we make a big point about helping to influence reimbursement decisions and to bring product cost of goods down to where they can be reasonably accommodated in the healthcare system, in the economics underlying the healthcare decision making. So one of the societies that we do that through is the International Society for Cell Therapy. If you want to learn more about it, just track back to the URL here, and I think this will be available following the meeting. And you can learn a lot about the initiatives that are going on there. One of the things that we recently did is to hold a dialogue with private payers, the insurance companies that make a decision on whether you are eligible to receive some of these emerging therapies. And we did it collaboratively with two other groups, an industry group, the insurance providers, and as well the translational physician community. And so just as a summary for what came out of that, it was really, really clear to us that this space is going to be driven to a low margin, high volume business. So we're not developing therapies as a company where we're going to get paid $100,000 for treatment and we're all going to see a huge amount of capital come back into the company. This is moving towards the need to make a $200 product that can be used broadly by a large number of people. So that's all good for the patient, and it's, from my perspective, it's all good because it puts the emphasis on being able to make a high quality product at large scale. So one of the difficulties with that is that technology is moving so fast in this space that the people that are locking in their commercial production process are being obsoleted by the next generation company. It takes something around 18 to 24 months from the time you say, this is how I'm going to manufacture a commercial product, to do all of the testing, to do all the repeat manufacturing runs, and then run your clinical equivalency study. And what I'm saying is during that time, new companies are coming up with new technologies that are leapfrogging over the early companies. So it's a really perverse environment where the pioneers that are launching the technologies are obsoleting themselves or being obsoleted just as a consequence of the timelines in the development process. So the other point that I want to make is that currently there are no requirements from the FDA to do comparative clinical studies. So we're making a product and it's similar to a product that Osiris Therapeutics is making. There's no requirement for us to do a clinical study and show that we're better or worse. And so what's happening is that the physician who potentially would prescribe a product doesn't have any information to make a discriminating decision. I mean, they have a label that tells them how many cells are in a bag from us or how many cells are in a bag from another company, but there's no, like on the back of a can of soup, you see like percent carbohydrate, et cetera, et cetera. There's nothing on these bags that says this has X amount of a necessary potency in angiogenesis or cytoprotection. There's no way for a physician to evaluate one product versus the other. And with what I just told you about manufacturing costs, this whole community is going towards a physician prescribing the cheapest product. So we've got to find a way through the FDA or through societies enforcing standards to get comparative data out there so a physician can make a guided decision that integrates costs with a comparative potency and also a potency in different disease settings. It's going to be a very chaotic environment with off-label prescriptions of cell therapeutics for disease indications they haven't been tested in, and a lack of information out there, even within my community, to make that a guided decision. So, this is going to be the end of the talk, and I'm very happy to take questions and dialogue following the talk. I had a very nice time at dinner this evening and met some people that I hope I can have some nice long-term relationships with. But I also realized that you guys have an interest in science in a very broad context, and you have a very diverse background. So it's been very difficult for me to, because I want to go into some very detailed science that I know is going to go way over your head, to let you understand the elegance and respect that I have for people that have developed out, much of the technology in this space. But I really hope that I could bring home a few points that would let you understand that medicine is changing dramatically, that there are ways from the patient side that you can help give input. You should watch the space. I just encourage you all to read and provide feedback through patient advocacy groups or ask questions through the NIH to help mature this business in the best, most effective way for the patient. And my last slide.
Thank you very much. So it's time for a few questions. There's a microphone going around. Please stand up and state your name and your affiliation with the society. And speak to the microphone. So the microphone is coming up.
Hello, my name is Rudy Kruger. I'm a member of the society. About a month ago, I went to bed and had my legs seized up like a Charlie horse. And it went away very quickly. It was very intense, went away quickly. And then the next morning, I woke up with a severe backache and my right leg was in pain. I went to my doctor and he sent me for x-rays, and I eventually got them interpreted by my orthopedic surgeon. And it showed that there was a compressed vertebrae, and the discs between them were bulging. And I want to know when I can expect to be able to be treated with these technologies.
Yeah, it's actually...
And if perhaps the fabrication lab that was described in a recent Philosophical Society lecture could be used to speed up the creation of the robotics needed to manufacture these things.
Yeah.
And also, the last question is, if I am treated, how long does it take for relief?
So let me, as your condition would continue, you would be headed for a surgery called spinal fusion. So it would be basically repairing the region of the bulging disc by coming in with a metal cage. And right now people use either what's called an allograft. It's bone that might be taken from your body but devitalized, or it might be cadaveric bone. It's basically just a physical scaffold that's used to repair that region. And so that has success, but it's not meeting clinical need. There are clinical cell products out there now that combine cell types that I've just described with you with that allo bone matrix that actually stimulate rapidly the formation of good bone, and you get a very good outcome from the spinal fusion surgery. And so those are companies like Nuvasive, and I'll give you my card afterwards and I can send you these companies, and you could go to if you were scheduled for the surgery, you could go to your orthopedist and you could ask him to evaluate those, but they would be available for him to elect to use. The other thing that people are doing is directly injecting these cells into the region of injury, but that's under clinical testing, so it's not something that your orthopedic surgeon could have you undertake currently. And so I can point you, for all of you.
How long before you can?
Pardon me?
How long before you can?
So you saw the cycle that I showed you of testing, which is generally so they're in phase 2 clinical testing currently, so you're probably talking 5 years before they would be commercially available. But one of the things all of you guys can do is elect to be treated in a clinical trial. And so the way you learn about those most effectively is going to a website called clinicaltrials.gov, and it gives you a searchable database that you can search for indication, for company, and you have refined searches, so you could look for cell therapy for disc repair. And it will show you all of the active drug or biologic or cell therapy trials that are currently enrolling, and it'll give you all of the inclusion criteria, how sick you have to be, and what specifics in your disease setting do you have to be eligible for the study. Take that to your physician and say, this is what I'd like to do. he'll refer you to centers that are active in the study, and you can see if you're eligible and participate. So you can do this with type 2 diabetes, stroke. You can do this with any disease that you want to evaluate. Go to clinicaltrials.gov, find out what's running, and then either go to the NIH and a patient advocacy group to get feedback on, patient experience, what's good and what's not, or go to your physician and ask them to evaluate it for you. But and again, if anybody wants, I can help you find the right links through patient advocacy groups to vet that. I mean, one of the points that was made in the introduction was this comment about medical tourism, and what we see quite frequently are parents children with cerebral palsy are desperate for treatment. And so they're doing the same thing. They're trying to figure out where they can go for treatment, and this buzz is stem cell treatment will cure all. And in fact, there are some very promising results coming out of Duke University using stem cells from cord blood to treat cerebral palsy patients. It's very encouraging data. But it's a small clinical study and doesn't meet the needs of all the CP population. So many people are going over to Korea or China. They're paying $50,000 to $75,000 for a cell treatment that's not yet been developed for optimal dose, and it's purely experimental. It's a surgeon or a physician just deciding, I can do this and we'll see what happens. And so it's very difficult for parents to get information about who's reputable and who's not, and what the experience is at a given clinical center. It's really critical that people are able to vet whether somebody is up to standard or not up to standard, even if different regulatory standards are used. There's a stem cell society called ISSCR, International Society for Stem Cell Research, that is very active in providing that kind of guidance for medical tourism to patients, to let them know what's reputable or not reputable at foreign centers, and to try to guide good decision making in that process. But it's really key that we limit the sort of non-evidence-based medical approach to developing these therapies. It's very important that the therapies are tested in controlled clinical studies, so you have a placebo group against a treatment group, that studies are done with deep enough statistics that you have a confidence level that something can work. The one-off practice by physicians is a very dangerous environment for this stage of development in this space. So
next question, back here. Hello, my name is Pierre Cartier. As a professional, I'm a dentist, and I've actually given some lectures on regenerative biology at the NIH through their graduate school regarding the dental applications. But I wanted to ask you, number one, what applications do you see for oral health in stem cell technology? And in addition to that, with regard to stem cell technology, how do you see using the immunomodulatory properties of stem cells in treating things like periodontal disease that have an etiology based in inflammation?
Yeah, very good. So right now there is a stem cell product that's being used in repairing injured gingiva from oral surgical procedures. It's coming out of a company called Organogenesis, and if you go to their website, what they're doing is adapting an ex vivo skin product to. He's got his phone out. He's checking me out. They've got a skin transplant product that they're using orally. And if it's not organogenesis, it's advanced biohealing. But there is a movement in this space to repair soft tissue from oral surgeries. The other place where people are using the technology, and we had run a phase 1 study when I was at Osiris Therapeutics, is in rebuilding the bone underneath a tooth that's been removed, whatever the appropriate term is. And so often it's difficult to get a post back down in and well seated in order to put a crown on top. And what we were doing is delivering a small amount of HATCP, hydroxyapatite tricalcium phosphate, embedded with cells immediately after injury and tooth removal, and it would stimulate bone repair. It would be 4 weeks later, a core would be pulled out of that, and then a post put in, and then the cell type used. People are pulling cell type out of the mouth from, you'll see it every now and then, from There's a gingival stem cell that's been isolated, and people are pulling it out from dental pulp from children whose teeth have been early removed, and then they're using the cells similar to the MSC type environment. With respect to inflammation in the periodontal space, I think right now it's a risk-benefit ratio. It's whether inflammation as a complication of dental surgery is of sufficient risk that it merits the cell therapy treatment. Because we have such short-term, insufficient 10-year follow-up for treatment of the cell type.
My name's Timothy Thomas. I'm a member. My question is very basic. As I understood, stem cells, they divide and specialize. But I'm getting the impression from your talk that these cells don't do that. They just die. So they're producing some kind of a protein or excreting something that's causing this effect. Why don't you just make the stuff and leave out the stem cell?
It's a great question, and it's often asked. And I think And I think that it's a undervalued component of this approach, is that by understanding more specifically the pathway through which things are acting and the biologic or combination of biologics that may work, you may eliminate the risk of a cell-based therapy and have a very effective biologic. And people are starting to do that. From some of the work that's been done using this class of cells in heart attack have shown that the cells that are getting lodged in the lung are secreting a biologic protein called TG16, and people have shown that IV administered post-heart attack can have a significant influence on reducing scar size. But what it's not doing is stimulating angiogenesis. So one of the drawbacks with the biologic approach is that you're only hitting a single modality. You're not getting these repair processes expressed in parallel. And so in the example I showed you with spinal cord repair, I mentioned that this methyl metalloproteinase could prevent axonal dieback, and it can. So we know that a small molecule or drug can stop these neurons from pulling away, but it's only in the presence of the biologics, the growth factors that our cell make, that we stimulate growth coming back forward. So I think in a simple pathology, it's a very good argument. I think many of the diseases we're left with unmet need are very complex and multivariate, and you need a more dynamic response. It's a very good question.
Stuart Reuter, member of the Society. This might be slightly off topic. Are you familiar with a treatment for brain injury called hyperbaric oxygen treatment?
Yeah. So
does that in some way stimulate local cells to do some of the processes we've been talking about? It's basically putting people in about a 2-atmosphere oxygen atmosphere for several times over a period of time. And there have been very good results in both PTSD and traumatic brain injury.
Yeah, I don't think people understand well the mechanism underlying the hyperbaric oxygen. I know it's also used in wound repair as well. But the linkage and the supposition that it's linked to stem cell or progenitor cell recruitment or function is quite good. One of the key sort of magic dust ingredients in our ability to expand these cells is growing them in a hypoxic environment by keeping the oxygen environment extremely low. Hematopoietic stem cells in the bone live on the endosteal niche, which is a very, very low oxygen tension environment. So it's very likely that what you're doing is influencing progenitor cell function by putting the area of injury into that hyperbaric environment. But I don't know more than that. To my knowledge, the underlying mechanistic cause and effect with that response is not very well known.
You know, for years it's been used against gangrene.
Time for 2 more questions.
You mentioned some of the dental applications, and I was wondering if you have, or if this technique could be used to cure osteonecrosis of the jawbone induced by, regrettably, osteoporosis.
There's the cells of this type have been very effective in focal repair of bone lesions. They've been used in multiple myeloma, lytic lesions related to disease and radiation treatment. And I believe that they have been used in osteonecrosis. I think the probability is extremely high.
So if there's a metastasis of cancer into the bone, it's also showing promise?
In terms of, first you have treatment to rid the bone of tumor. So coming back in with these cells on a matrix to repair, or injecting these cells, and many of the matrices that these cells can be applied to are almost like putty, or are syringe injected. You can inject into an osteonecrotic site, cell plus bone, cell plus scaffold, and see significant bone regrowth.
And if John will forgive me, can I add a little?
Well, let's. Sure, maybe I can ask the speaker after. Let's get one more question here, the last one.
My name is
Oh.
Ronnie. Oh, I'm sorry.
Okay, go ahead,
My name is
sorry.
Ronnie Perry, I'm a visitor, and I have a question about whether stem cell work is actually being done in this country right now for conditions like osteoarthritis and rheumatoid arthritis, and their after effects, such as the need for a hip replacement or knee replacement,
Yeah,
that sort
so
of thing, and where these treatments might be currently.
So I'm sure that there are phase 2 studies using cells of this type in rheumatoid arthritis, and they're either IV infused or injected into the joint. And the rationale is that you can block the immune destruction of joint tissue. So it's really, but it's not a treatment to remodel or repair the joint, but it's a treatment to block the activity. So to my knowledge, the cells have not been used very effectively in osteoporosis. And part of that is that for the cells to regenerate new bone, they're subject to the signals in that tissue environment.
Osteoarthritis.
Osteoarthritis is a plus. If you went to clinicaltrials.gov, you'll find clinical studies going on, and cross-search for cell therapy, you'll find clinical studies going on using cells of this class, both for bone repair but to limit immunomodulation. There's, people have directly injected the cells into the synovial space in patients with meniscal injury or osteoarthritis and seen stimulation of soft tissue that cushions and limits the pain that's seen, if not corrected some of the damage from the osteoarthritis. There's a study from Athersys Therapeutics, and there are several publications out about that.
You mean cartilage? Replacing
Correct.
cartilage?
So not directly replacing cartilage, but replacing the soft tissue that can cushion the cartilage that's been worn down, so you avoid a bone-bone contact. So, and again, if you guys can give me an address after this, I don't have the answers, but I know where to send you to get more information. I'd be very happy to help that way.
Thank you very much. The speaker will be around during social hour to answer more questions. In appreciation of your lecture this evening, we have the announcement framed and signed by the members of the General Committee for you to keep.
That's very nice. Yes. This is really nice. Thank you. That's very nice.
Thank you.
My appreciation.